I'm John Strum, and this is Real Talk MS. It's April 28th, and we have a lot to talk about. As many of you already know, myelin is the fatty substance that protects the nerves in your brain and spinal cord. And MS is called a demyelinating disease because in MS, the immune system mistakenly attacks the body's own myelin. The damage caused by those autoimmune attacks leads to MS symptoms and disability. Now, myelin repair is a natural function. But for people living with MS, that ability to repair myelin eventually stops working. New research led by my guest, Dr. Larry Sherman, may explain why that happens and point to new possibilities for future myelin repair treatments. Dr. Sherman is a professor at the Oregon National Primate Research Center at Oregon Health and Science University, and his research focuses on how the brain and spinal cord respond to injury and disease, particularly in demyelinating conditions like multiple sclerosis. But before we get to my conversation with Dr. Larry Sherman, there are a few other things that you should know about. Last week, the European Medicines Agency's Committee for Medicinal Products for Human Use recommended the approval of tolebrutinib for treating adults living with non-relapsing secondary progressive MS. Tolebrutinib represents a new category of disease-modifying therapies called BTK inhibitors. The BTK stands for Bruton's tyrosine kinase, which is a very long name for a protein that plays an important role in the development of B cells. A BTK inhibitor blocks the activity of this protein. And one thing that makes tolebrutinib particularly important is that it can cross the blood-brain barrier, so it can target B cells in the central nervous system. Now, some approved DMTs have been shown to delay progression following a relapse, but tolebrutinib has been shown to delay disability progression independent of relapse activity. And that progression independent of relapse activity, often referred to by the acronym PIRA, is something we often hear about. It refers to the worsening of MS symptoms and disability that occurs when someone isn't experiencing active inflammation or new lesions in their brain. And because this progression occurs in the absence of active inflammation or new lesions on MRI, doctors can only acknowledge it retrospectively, that is, after it's already occurred. So this should help you understand why many people are excited about tolebrutinib. It's particularly good news for people living with secondary progressive MS throughout the European Union. Unfortunately, the same can't be said for people living with secondary progressive MS here in the United States because this past December, for reasons of their own and contrary to all indications to that point, the FDA refused to approve this same drug for use in the United States. I'm of the opinion, and I want to underscore the word opinion here, that tolebrutinib should have received FDA approval. Clearer heads seem to prevail within the EMA's committee, or at least heads that were more clearly attuned to the needs of people with secondary progressive MS. If you'd like to hear more about what happened and perhaps why it shouldn't have happened, I'll invite you to listen to my conversation with Dr. William Conti, one of the principal investigators in the Phase 3 clinical trial that analyzed tolebrutinib and non-relapsing secondary progressive MS. You'll find that conversation in episode number 440 of Real Talk MS, and you'll find a link to that episode in today's show notes. We'll continue following tolebrutinib as the European Medicines Agency is expected to render its final decision on approval in the coming months. And while we're discussing BTK inhibitors, last week, Roche announced that it was submitting its BTK inhibitor, fenebrutinib, to the FDA and EMA for approval. In clinical trials, fenebrutinib was shown to reduce disability progression among people with primary progressive MS and relapsing remitting MS. In the clinical trial focused on fenebrutinib and relapsing remitting MS, fenebrutinib demonstrated an annualized relapse rate of approximately 1 relapse every 17 years, which is the lowest relapse rate ever observed in a phase 3 clinical trial in MS. In the clinical trial that focused on fenebrutinib and primary progressive MS, fenebrutinib outperformed Ocrevus in reducing disability progression, potentially making fenebrutinib the first and only high-efficacy oral, brain-penetrant treatment for relapsing forms of MS and primary progressive MS. Now, considering the fate of the last BTK inhibitor that the FDA weighed in on, we're keeping a close watch and making no predictions on what may happen when they analyze the clinical trial outcomes for fenebrutinib. So stay tuned. Disease-modifying therapies work, and as a result, people with MS are living longer. In fact, the majority of people living with MS in North America and France are over the age of 50. And as a result, people living with MS are finding themselves dealing with other age-related health conditions. Studies focusing on MS and other health conditions, or comorbidities, have typically looked at the impact of managing cardiovascular, autoimmune, and psychiatric conditions along with MS. But there hasn't been much research focused on the impact of a cancer diagnosis on MS care. So a research team looked at the impact of cancer on MS care and the use of MS disease-modifying therapies once an individual with MS has been diagnosed with cancer. The team used population-based data between 2009 and 2021 from France and between 1991 and 2020 in British Columbia, Canada. From this data, they identified 20,706 people living with MS, and of these, 4,555 people in France and 2,347 people in British Columbia received a new cancer diagnosis. Each of these individuals was matched with 2 cancer-free people with MS, and they were matched by the year they were diagnosed with cancer, their sex, their birth year, the area where they lived, the time since their MS diagnosis, and their MS disease-modifying therapy use. So we have almost 7,000 people with MS who had been diagnosed with some form of cancer on 2 separate continents in 2 separate healthcare systems, being compared to a control group of 13,804 people with MS who had not been diagnosed with cancer. And here's what the researchers found. Rates of visits to the neurologist were similar in both Canada and France and didn't differ in the 2 years before and after a cancer diagnosis. The rates of MS-related hospitalizations were low in both regions and did not differ between the people with MS who had cancer and the people with MS who did not have cancer. However, the use of MS disease-modifying therapy dropped sharply after a cancer diagnosis, especially among those people receiving chemotherapy to treat their cancer. What we don't know from this study is what kind of impact stopping DMTs had on the MS outcomes. So there's more research that needs to be done here because as the MS population continues to age, developing evidence-based guidelines for managing MS while an individual is also managing cancer will only become more important. Now, if you'd like to review the details of this study, you'll find a link to the published study results in today's show notes, where you'll also find a link to my friend Sharon Roman's excellent plain language summary of this study. Last Thursday, Heisman Trophy winning quarterback Fernando Mendoza officially became the number 1 pick in this year's NFL draft when he was drafted by the Las Vegas Raiders. Now, it's traditional for the number one draft pick to be present at the draft and stand with NFL Commissioner Roger Goodell when his name's called. But Fernando's mother, Elsa, lives with MS. And because she requires a wheelchair for mobility, travel becomes more difficult. So Fernando chose to stay at home and celebrate being this one's number one draft pick with his family. Also on Thursday, Fernando announced that he was partnering with the National MS Society in establishing the Mendoza Family Fund. And he made that announcement by making an initial contribution of $500,000, which puts the Mendoza Family Fund comfortably on track to reach its goal of raising $1 million over the next 3 years. If you'd like to learn more about the Mendoza Family Fund, or you'd like to show your support by making a donation, you'll find that link in today's show notes. As we have frequently discussed on this podcast, artificial intelligence is impacting every facet of healthcare, from the lab to the bedside. According to a recent report, more than 40 million people ask ChatGPT healthcare-related questions each day. Just a couple of months ago, OpenAI, the company behind ChatGPT, conducted a survey that found 3 in 5 adults in the U.S. had used an AI tool for health or medical questions in the previous 90 days. 55% of the survey respondents indicated they used AI to check or explore symptoms they were experiencing themselves. 48% used AI to understand medical terminology. And over 40% turned to AI to learn about treatment options, all of which explains why, just a couple of months ago, OpenAI launched consumer-facing ChatGPT for health. Well, now your doctor can also turn to ChatGPT, because just last week, OpenAI announced the launch of ChatGPT for clinicians, an AI tool designed for individual clinicians whose hospitals or clinics don't offer a centralized AI tool for them to access. Now, ChatGPT for clinicians is free for physicians, nurse practitioners, physician assistants, psychologists, and other licensed clinicians in the United States. And this is important. Each ChatGPT for clinicians response is evidence-based, including citations to journals, including authors, publication dates, and titles for full verification. ChatGPT for clinicians can answer a healthcare provider's questions, help provide research answers in seconds, and can also be used for common clinical workflow applications where ChatGPT can learn reusable skills to expedite things like prior authorizations, patient instructions, and referral letters. AI is changing everything, and we'll continue doing our best to keep you updated on what's next. Speaking of keeping you updated, I want to remind you that this episode of Real Talk MS is sponsored by Able Now, a tax advantage savings program for people with disabilities. If you're living with multiple sclerosis, this is important news. Expanded federal rules mean more adults with disabilities, including many people with MS, can open an Able Now account. Able Now lets individuals save and invest money without affecting their eligibility for certain public benefits like SSI or Medicaid. For many of you, it can be an essential financial tool. To learn more and understand if you're eligible to open an account, visit ablenow.com, and you'll find that link in today's show notes. While AI can be a great tool, it still takes great researchers to make the kinds of breakthroughs that bring us closer to solving the riddle of MS. My guest, Larry Sherman, and his team have made a discovery that could explain why people with MS eventually lose the ability to repair damaged myelin. And they may have also found a solution, which could be a game changer when it comes to remyelination. In a moment, we'll meet my guest, Dr. Larry Sherman. Myelin is the fatty substance that protects the nerves in your brain and spinal cord, and MS is called a demyelinating disease because in MS, the immune system mistakenly attacks the body's own myelin. While myelin repair is a natural function, it eventually stops working for people with MS. New research may explain why and point to new possibilities for future treatments. Joining me today is Dr. Larry Sherman. Dr. Sherman is a professor at the Oregon National Primate Research Center at Oregon Health and Science University, and his research focuses on how the brain and spinal cord respond to injury and disease, particularly in conditions that involve demyelination, like multiple sclerosis. Welcome back to the podcast, Dr. Sherman.
Thanks, John. It's great to see you.
I like to begin these conversations by getting our definitions in order, so I hope you'll start us off by explaining what myelin is and the role it plays in MS.
Sure. So myelin is a substance that actually wraps around nerve cells, and it has a lot of different functions, one of which is to protect those cells, the nerve cells. But probably the more important function, especially relevant to MS, is that myelin increases the speed at which nerve impulses travel through those nerve cells. So I always like to talk to people. I lived in Germany for a while, and the Autobahn in Germany has no speed limit. And so I always like to tell people that if you have myelin, it's like driving on the Autobahn at midnight where you can be going 200 miles an hour and no one's going to stop you. And if you don't have myelin, it's like driving on the busiest highway in the United States at rush hour, where you're lucky to be going 2 miles an hour. And that's about the actual change in speed. Myelin is so important because if you think about what our brain is, it's a computer, and it has to be a very fast computer. So all these different parts of the brain have to talk to each other, also talk to our spinal cord and reach out to the rest of our body. So having myelin is critical for that. And if you lose myelin, all of that goes away. So in MS, your immune system, for reasons we still don't entirely understand, decides that myelin is foreign and attacks it and strips it off of the nerve cells. And now you're going 2 miles an hour instead of 200 miles an hour.
So once we're stuck in that gridlock going 2 miles an hour, what types of symptoms might someone develop as their myelin becomes damaged?
Yeah, so just about every symptom we see in people with MS is a result of losing that myelin. So it's very important for your motor functions, your ability to move. It's very important for sensory functions, your ability to feel things and sense things, and also can cause pain, for example, because of misfiring. And then finally, it's really important for cognitive functions. Our ability to learn and remember things can be impacted by losing myelin. So just about everything our brain does can be impacted by myelin loss.
You know, as I mentioned when I introduced this segment, myelin repair is a natural process, but over time, that process stops working for people with MS. Do we know why?
We have some hints, and my own research suggests that at least one of the factors is a change in things that are happening outside the cells that make myelin over time. And so you're right. In early stages of MS, we think there's a lot of remyelination happening. The cells that make the myelin come back and they make more myelin, and everything's usually okay. There's a second process that happens, and that is that it turns out throughout our lives, and this is kind of relatively recent data, we have these cells in our brains that are called progenitor cells. And these cells, when there's injury to myelin, kind of move over to where the injury is, and then they turn into the new myelin-forming cells. They're called oligodendrocytes. And so these cells make myelin, and these other cells make myelin-forming cells. And so over time, what we're finding is that we get this kind of pileup of these progenitor cells that should be making new myelin-forming cells, but they're not. They're stuck. And so we've been trying to understand some of the signals that prevent that from happening.
Your research has highlighted that the brain often tries to repair itself, but gets stuck. Can you tell us more about that?
Yeah. So in our research, one of the things that we figured out that's preventing remyelination is a molecule, or an enzyme, I should say, called CMYP. CMYP stands for cell migration and hyaluronic acid binding protein, which is a lot harder to say than CMYP. So CMYP is an enzyme, and what it does is it chews up a molecule that you probably all heard of called hyaluronic acid. It turns out that hyaluronic acid isn't just for facial creams, which is where most people hear about it. Hyaluronic acid gets made whenever there's damage in any part of our body, including the brain. So after these assaults on our myelin by our immune systems in MS, hyaluronic acid piles up, and it's probably good at first. It's probably limiting the inflammation. But over time, when there's lots of MS attacks, it starts to pile up to excess. There's too much of it. CMIP comes along. It's being made by these cells that turn into the myelin-forming cells and starts chewing that up to degrade it and get rid of it. But if there's too much hyaluronic acid, the little pieces of hyaluronic acid that are made by that enzyme feed back on those cells and tell them not to become myelin-forming cells. And so if we could attack this enzyme, that might be a strategy for myelin repair.
I was about to ask, how might targeting CMIP influence future treatment for someone living with MS?
Yeah, so we've been spending the last several years trying to develop a drug that stops this enzyme from doing its job. And we've now come up with several drugs that are good experimentally. And what we found is that if we stop CMIP activity during demyelination, it actually promotes and accelerates remyelination. And we actually start to see some of the neurons, they're not back on the Autobahn exactly, but they're going about 75 miles an hour, and that's pretty good. And we see some functional recovery as a result of that. So right now we're trying to find better drugs that really limit this effects of this broken down pieces of hyaluronic acid by preventing them from being created in the first place.
If we can successfully remove those biological breaks that prevent remyelination, what might that look like for a patient? Do you see, and I think you've already started to answer it, whether it's a reversal of their symptoms or maybe just a stabilization of where they are?
So we've actually tested that question in both mice and also in a non-human primate model of MS. So in mice, we see really good functional recovery. The problem with any kind of strategies using that are really trying to repair the brain in mouse studies is a mouse brain is only that big. It's really tiny, and it's really physiologically very different than our brains. My lab is out at the Oregon National Primate Research Center, as you mentioned. And the reason I came here was because back in the 80s, we had a group of Japanese snow monkeys that spontaneously developed a disease that looks very similar to multiple sclerosis in humans. In fact, we think it may even be caused by the same things that cause MS in humans. And so we've taken the drug now to these animals that have demyelination in their brains and spinal cords. And so far, the drugs we have been able to show remyelination in those brains as well. But we're getting them at a stage of disease that's pretty far along, and the drugs are not optimal yet. They're not quite perfect as drugs. So we can see remyelination happening, but now we got to go to the next step and see if we can get it to actually promote recovery, bring back motor function, bring back the sensory problems, and actually improve cognitive function as well.
You just said something that I find fascinating. You said these Japanese snow monkeys spontaneously developed MS. So this was not something that was induced in a lab. And so the work you're doing actually has the potential to cure them.
That's what we're hoping. My whole reason for coming to the center was to cure these monkeys, because I figure if we can cure these monkeys whose brains are much more like ours than the mouse brains, we'd have a really good chance of having something we could take to humans. The other thing these monkeys may tell us, though, is how MS starts, which I think would be incredibly exciting. We think it's triggered. We have good data showing that it's triggered by a combination of a genetic liability that's similar to genes that we know are affected in MS patients, and a virus that arose in that colony back in the 1980s. And the virus is very similar to a virus that's been linked to MS, Epstein-Barr virus. So we think that on the one hand, they're great for trying to figure out how to fix the brain. On the other hand, they may tell us how to stop MS in its tracks.
You've spent years at OHSU moving concepts from the lab toward clinical application. What's the biggest hurdle in getting a promising myelin repair molecule through human clinical trials?
We've seen a lot of labs that have come up with different strategies to promote remyelination, and that work is ongoing. Just beautiful work from lots of different people. And I think we're all sort of converging on similar ideas in terms of what pathways and cells we want to go after, including from hyaluronic acid and CMIP. The challenge is to get, again, I think, going from these model systems into the human brain. And that's one of the things that I'm really excited about having this monkey model, because having the monkey in the middle really gives you an opportunity to see if this will work or not before taking it to the clinical trials. The trials that we've seen so far have been not exciting, really. I mean, there's some excitement around some of the things that people have been able to see, but they're not quite where we want them to be yet. And so I think having better platforms for testing this before we take it to people is going to be a really important thing to overcome.
It's occurred to me that while we're talking about the mechanics of myelin repair, I'm wondering how brain atrophy impacts the ability to induce myelin repair.
So that is a challenge, right? Because the problem is if the axons, which are the little threads that myelin wraps around on the nerve cells, if they're gone, then there's nothing to remyelinate. And as we lose nerve cells, we're going to lose the ability to promote remyelination, obviously. So I think for progressive stages of disease, the really important thing is going to be combining strategies for remyelination with strategies for neuroprotection. And what I mean by neuroprotection is saving the nerve cells themselves, the neurons. And so there's a lot of people studying that right now. And there's some really promising work coming out of multiple labs looking at neuroprotection. So if you have now something that's going to promote neuroprotection and then something else that's going to remyelinate those axons that are protected, then you start to have real possibilities for the more progressive stages of disease.
You and I actually last spoke nearly 8 years ago in episode number 67 of this podcast. So if we were to sit down again in another 8 years, what's one breakthrough in myelin repair you're most confident that we'd be discussing?
I think in 8 years, I don't have a crystal ball, and so this is completely speculation. I think in 8 years, we're going to understand so much more about the molecular basis for how myelin works and how myelin is made, and then how myelin comes back, that we're going to have so many better strategies for myelin repair than we have now. The thing that I think we now know is it's possible. And if you'd have asked me that 20 years ago, I would have said, I don't know, right? Because we just didn't have the data to suggest that was true. And now we know it is possible. All the experimental data from all these models that we've been testing show you can promote myelin repair. So now the question is, we need to get all this data together and find exactly the right way to do that. And I think in 8 years, we're going to have a much better idea of what the right strategy will be.
Well, Dr. Larry Sherman, I want to thank you for all you and your Japanese snow monkeys do to effectively improve the lives of people living with MS. Thanks so much for talking with me today.
Thank you, John. Great to see you.
That's going to wrap up this episode of Real Talk MS. Real Talk MS is powered by the National MS Society, and you can share this episode of the podcast by letting your friends or family members know that all they have to do is point their web browser at realtalkms.com/452. You'll find that link in today's show notes so you can easily copy and paste it right into an email or a text. Next week on Real Talk MS, my guest, Dr. Riley Beauvais, tackles one of the most important and personal topics for many women living with MS, the intersection of MS and the journey to parenthood. Navigating family planning with a chronic condition can sometimes feel overwhelming, but Dr. Beauvais brings world-class expertise and a reassuring perspective to the conversation. We'll be discussing preconception planning and how to manage disease-modifying therapies when you're ready to start a family. We'll look at why women with MS often see a reduction in relapse activity during pregnancy, and we'll review practical strategies for managing the increased risk of relapses after delivery. Whether you're currently planning a family or looking to support someone who is, I hope you're planning to join me for next week's episode of Real Talk MS. I'm John Strum. Thanks for listening. Stay safe and make healthy choices.